srpnju. So I'll switch to English now because I'm just more used to talking about this in English. So the mission is launching in July 17, and it's a rover mission to crater on Mars that is called Jezero, or Jezero as they all say here. And I stopped correcting people because I don't want to be the one who is like, it's Jezero. It's after the Bosnian town called Jezero. So this mission, unlike any previous mission to Mars, has an unprecedented goal, and it can be historic in that its objective is to go to this crater. We'll see a lot of this picture in the talk that contains really evidence, visible evidence from space of former water activity and former sedimentation. So there was a lake in this crater once upon a time, 3.8 billion years ago. And the mission, the rover, will identify samples that are most likely to address a variety of goals and pick them up, cache them, and eventually these samples will be returned to Earth to be analyzed in all sorts of terrestrial laboratories to address various questions. So some of the major mission objectives are here. So to date, we have no samples from Mars that are from a known geologic context. And this would be the first time to do that. So here are the mission objectives. The first one, just like any other mission, is geologic exploration. So the rover will go to Mars and will explore this ancient environment on Mars. I said it's about 3.8 billion years ago that the crater and the lake were present. Now it's dry. And just by looking around and trying to understand the sedimentology of the place, we can already start understanding processes by which the sediments formed, by which different minerals in these sediments formed and were altered due to interactions between water and minerals that were maybe originally present. Another goal, and this is a goal that has never been really stated by NASA before as related to any Mars mission, is to look for biosignatures. So habitability, the potential of an environment to be inhabited by life, has been the goal of many previous missions, including the currently operating mission in Gale Crater, another rover mission, which has assessed habitability of that environment and has demonstrated the former presence of water and sedimentation in that environment. So you probably have seen many news articles that report yet again water on Mars. So that is almost not a question anymore. We know there has been water on Mars, and we know there have been habitable environments. So this encouraged NASA to, for the first time, state that now that we know there have been, or there were habitable environments in the past on Mars, let's try to seek evidence of past life, because these environments are incredibly old. They're older than any sedimentary rocks, and they're better preserved than any sedimentary rocks that we have on Earth of similar age. And so let's just go, and this is where people like I come in, participating scientists or sample return scientists who will try to figure out what the best samples are to pick by the rover and then bring back to Earth and look for biosignatures. So any kind of sign of life or former prebiotic chemistry potentially. So a major goal, unlike any previous rover, this rover, Perseverance, just recently named, will prepare a returnable cache of these samples. About, it says here, collect about 40 samples. There will be really 30, as well as some blanks, procedural blanks, witness plates, and so on, that really sample diverse geology within this crater that can help us eventually address all sorts of questions. So the rover itself has limited capacity to investigate these samples. We can say something about the minerals, we can say something about the sedimentology, but all sorts of analyses that take place on geologic samples from Earth can take place once these samples come back to Earth. So really small scale, really high sensitivity analysis. And another goal here that you see is to of this mission, just like other missions before, is to prepare for human exploration. So there will be some measurements of temperature, humidity, just the weather, as well as the testing of MOXIE, an instrument that needs to demonstrate the ability to convert CO2 from Mars, the Martian atmosphere, to oxygen. So here is just the notional idea of how this will work. So here's if the rover lands on this. Can you see my cursor? I hope you can see my cursor. So this X is where the rover lands. It'll drive around. If it sees some region of interest, it can look around. We will explore it. We will try to understand what happened there. And if we notice some samples that can help us address different questions, scientific goals, then we may sample that that environment. These are sample tubes. You can see these, they're really pencil thin. So these samples will not be large, but a lot of geology and geochemistry on Earth is done using samples that are not much bigger, as well as a lot of planetary science using meteorite samples. So these sample tubes will be collected and deposited in one place, multiple tubes in one place, that then will be picked up by successive missions. So not this particular mission, there will be another mission that picks up the tubes, and then another mission that picks up these tubes from the orbit around Mars and brings them back to Earth. And that's supposed to happen. The date is not still set in stone, but by maybe 2028 or really 2030, realistically, we may have these samples back. So here's the rover. The rover is, it may look familiar. It is really the iteration of the Curiosity rover that's currently in Gale Crater. There have been some updates to the wheels. So here's the size. You may hear often that it's about the size of a Volkswagen Bug beetle. And here's what it looks like. It has various instruments. It has extendable camera arm. It has a drill, so we can collect samples of rocks and sediments. And there are various instruments on it. Although the suite of instruments that is present, really, as the primary goal of this rover is to collect the samples, there are not so many instruments to really analyze samples in situ. The whole point is to bring them to Earth. So here are some of the instruments. It's really a complex system, and there are all sorts of, so there is the ground penetrating radar. You can see all these flags show you that there are international collaborations. So Spain is contributing some wind sensors, some atmospheric measurements. France is contributing SuperCam, which is an instrument that takes pictures, photos of rocks, as well as can do some analysis to tell us whether maybe silicon is present or calcium, so we can have more insight into the elemental composition. Norway is contributing the ground penetrating radar. And so there are people from all these countries working on the team. And most of this, really, the rover itself is was assembled at JPL, and was just delivered maybe last month to the Kennedy Space Center in Florida. And out of the instruments to analyze rocks, so you see there's quite a bit of stuff, out of instruments that are really of most interest to someone like me and geologists, we have, well, first our eyes, SuperCam. It's a SuperCam. We have Mastcam Z, which is really the same kind of camera that you use to getting images from, if you've seen any images from Gale Crater, from Curiosity, it's really the same system. So these are our eyes. there are instruments such as Sherlock and Pixel, which have the ability to produce inabraded rocks as well as unabraded rocks to give us insight into the elemental composition as well as detect some organics. So there is Raman on among these instruments, the Raman spectrometer, so we can see whether maybe there are organics. It's also sensitive to specific minerals that we can look for. So we'll know something about the spectral signatures of minerals that are present, which is really, as you'll see later, this will be really important when we look for the best possible samples that could preserve biosignatures, if any were present. And here's the timeline. So here's the launch date. Usually there's a window around it. So nominal launch date is on July 17. There could be a couple of days, just like this most recent launch, the SpaceX launch was delayed just because of the weather. There are a couple of days we as the team were supposed to go to Florida and watch it. Unfortunately, because of coronavirus, it will all be online except for the launch itself. So all of the team scientists will be watching it online, which, well, I had higher hopes for this summer. And the rover, we know exactly the date when it's supposed to land on Mars, and that date is February 18, 2021. So that's just half a year, a little more than half a year to get to Mars. And the surface operation. So once the rover, and it's really the same landing system as the Curiosity rover. So we are using existing technology with the change of really caching samples and having sample tubes. And the surface operations are supposed to, so once the rover lands and everything is checked and everything works, it's supposed to operate on surface within the Jezero crater for at least 1 Mars year. And that's about 700 Earth days. That is, as you may have seen from Gale Crater, these rovers tend to last a long time. So this is very conservative. This is what you'll hear from the mission directorate, but these rovers tend to work for much longer. And so there are plans for an extended mission that would go out of the crater and maybe drive for a year or two to get to another interesting area. So those are tentative plans where we could even collect additional samples. So these things go on for years. So it could be more than just 1 Mars year. And so what I'll focus on today, there are clearly hundreds of scientists working on different instruments and about 10 of us who are return sample scientists who are really in charge of recognizing the best samples to answer specific scientific questions. I will focus on, you could focus on anything, Mars weather, you could focus on geochronology, so how old are different rocks and when did certain processes happen. What I will focus on today is a set of questions that I think samples that are sedimentary rocks from Mars can really help us answer, which is why the mission is going there in the first place. And this is the set of questions. So did prebiotic synthesis occur in Mars? Was there life on early Mars? And how, when, and why did Mars change? And so we'll go into these questions and try to give you a little bit of background and why this is really an unprecedented mission that can answer some of these. So as I said before, it's no news to pretty much anybody that there was more water on Mars in the past. And people started realizing that even from Viking orbiter images. So this is in the 70s. A lot of people were probably not even born there then. And so here's just a photo from the orbiter, and you see a lot of craters on this part of the really old surface of Mars that have river networks. And so even looking at Mars from space, one can see that water was flowing on the surface way back when. Now all of that is dry. And so to place Jezero crater is another of these craters. To place Jezero crater in context, so here's just the Mars geographic map. And what you see in colors, so the blue colors just show you lower areas, and the northern hemisphere of Mars is generally much lower, kilometers lower. This is in meters, many kilometers lower than the average topography of the southern hemisphere. And there is this area, Nili Fosse, that is the boundary between this really old high terrain and the lower terrain. And one of these little tiny craters in this area is Jezero crater. And the reason, there was a lot of debate where to go. It's a $3 billion mission, so choosing the landing site is very important and is not an easy task. There were many places considered. And I went to a couple of landing site workshops. So there are workshops where the community can come and make their case and present the case for different places and state the reasons and excite people about the potential science that can be done. And so here's why Jezero crater won over all these other sites. I think there were 40 or more in the initial rounds. So this is what a crater looks like from space. It's about 50 kilometers in diameter. And it is, as I said, let's go back to this, it is between this really high terrain and the low terrain. It's in the old, so all of this higher terrain is older. Geographically, it's older. We know that from crater counting statistics, there are just a ton of craters on top of craters, meaning there has been a long time of impacts, whereas the northern hemisphere is just not so cratered, meaning it's younger. So this crater is in the area that contains some of these river networks, so evidence of former activity where rivers, so liquid water, was moving on the surface of Mars. And here's what excited people a lot. You see this vein here. So we have the topography generally goes, this is higher, the crater itself clearly lower. And the water was flowing like this and depositing this vein. So it is this vein that has unambiguous signatures, and this is all based on the photos from space. It is unambiguous that this was a delta. So you have fast flowing river that carries sediments and then drains into a standing body of water, which is where deltas form. And so we have a standing body of water, great for habitability if there was any life, and sediments that are being deposited that can also preserve all sorts of biosignatures. And it was really hard to debate the value of that place once that was demonstrated. It took a lot of imaging from space, and it took a lot of orbital spectroscopy to understand the types of minerals that are present. And we'll talk about some of these minerals. So this is why Jezero crater was chosen, and you can see here, so this rim is a little bit eroded here, and you see even some much smaller craters within. So that is these are very tiny, but overall, just dating the terrain based on crater counting, which is calibrated by the moon, it seems like the crater and the lake was present around 3.8 billion years ago, which is, again, as I said, older than any sedimentary deposits that we have well preserved on Earth. So that alone, and here you see these sediments that actually look way better preserved than a lot of maybe 1 billion year old deposits on Earth. So even just looking at what this aqueous activity and river activity was like almost 4 billion years ago is amazing. Another aspect of going to this place is we don't know how long the lake lasted, but we know that it was filled almost to the rim There is no evidence for any kind of desiccation. There is no evidence for any kind of periodic level drop. But what we do know is that the lake overflowed, it got completely filled up, and then this additional channel opened up, and this is where the lake was draining. So it seemed like an open system, water coming in here, water going out there. So that is this outflow channel in the lake, and this is the delta. And this is really the area where the landing site will be. And so we have a lake, we have a delta, we have sediments, all of these different colors in this image. I'll be showing this image a lot, so now you have the context. All of these areas have been mapped mineralogically. This is what some of these colors reflect, different types of minerals that are present. So this is the inflow channel, this is the delta, and this is this old volcanic basement. So this is higher. So the water is going from that high place to the lake. It was flowing to the lake itself. So from this volcanic basement to the lake, and there are layers or there is just what we know from spectroscopy. There is quite a bit of olivine carbonate present in the lake. And I will explain the significance of that, or the potential significance of that, if you don't know what olivine is, and I certainly did not when I graduated from physics. It is really one of the most dominant minerals on Mars. It is produced by the volcanic activity and is present all around in this volcanic basement. So in the entire area around the lake, there was olivine present, and the origin of carbonate is a little less clear. And here's this vein deposit that contains olivine carbonate as well as a lot of clay minerals. And then there's this fairly flat floor, and there's quite a bit of debate as to what that floor is. Is it volcanic? Is it some kind of just deposit that maybe even later than the vein itself? So just by landing there, we can start addressing these first order geological questions. What are we looking at? So more about the minerals in the delta, which is a major argument towards former habitability of the lake. It tells us something about the pH conditions that were present, tells us that the pH was not something insanely low. It was not probably very high either. So the information about the types of minerals in the delta so far has been derived from orbital spectroscopy from other missions. And this is what the data can look like. So this is where physics comes in. So people, by having a database of different minerals and their infrared, this will be reflectance values, can recognize certain mineral signatures. Some minerals are better or more recognizable than others. And by acquiring spectra, so these spectra are acquired with about, depends, like 20 to 30 meter per pixel resolution. And by looking at the spectral signatures, if you see some in particular, these absorption dips, you can see that in some areas, these two dips are larger, which is the areas with carbonates. So in particular, magnesium carbonate, there will be some areas where there may be smectite, a clay mineral, and there's really all areas contain a strong olivine signature, which will be really more of these dips here. So all of these areas have olivine. Some just have less of other stuff. And so what is olivine and what are all these minerals and why does it matter? So olivine is really, it's not a mineral itself. It's a bunch of different minerals that contain magnesium and iron. It's silicate minerals that are produced by volcanoes. So there's not too much aluminum. And as I mentioned, olivine is really abundant everywhere in this high area around the lake. If olivine reacts, once it's extruded, olivine is what you find in basalts on Earth. So for example, I think is it Sveta Jabuka, one of our islands is basaltic. And olivine is not really stable in the presence of water and surface conditions, and it will react with water. If there's CO2, it'll react quite readily to produce a suite of minerals. So these minerals can be clay minerals. There could be various, I won't name these minerals, doesn't really matter. There are spectral, there are indications. These are much harder to recognize as specific minerals from the spectral signals, but there is potential evidence for just silica dioxide amorphous. There is spectral potential evidence for this and that, and this is serpentinite talk. Anyway, there can be a lot of different minerals present. These clay minerals are products. This is important. Here's where the water comes in. Water reacting with olivine makes all of this. And what we do know also is all of these kinds of minerals, a lot of these types of minerals are also present outside of the crater. So there was some broader scale, more regional scale activity that combined water with olivine and made clay minerals. So the question now about clay minerals in the crater itself is, were they produced in situ right there because the lake water was there? Or were they just brought as this river was draining and bringing sediment particles to create this morphology, geomorphology here? Were these minerals just delivered from some areas further up? These reactions with water, if there is also CO2, can also make carbonate minerals. So there's magnesium, there's CO2 under certain conditions. We can also produce magnesium carbonate, which is actually called magnesite, and that is this green area here is enriched in this particular mineral. Again, some of that is present outside of the lake. It is very intriguing that this green area in the lake itself is stratigraphically restricted. It's really coincides with a high water level. So it almost makes one think of a bathtub rim. So some of the carbonate could have precipitated in the lake, reflecting a very specific lake chemistry. So this is where we'll land. This is the landing ellipse here. It's sorry for the overlay. It's a little fuzzy, but there's usually missions get a landing ellipse. So anything within the ellipse is good. And we aim for just landing within the ellipse somewhere. And the idea is from the ellipse, we will start moving. And this depends on the trajectory itself, really depends on a lot of engineering and the understanding of really orbital imagery that tells us you should not drive into a dune field because there is some sand in there. And so we'll move from the landing ellipse over the delta, hopefully sampling some areas with carbonate, some areas that also have enriched signatures of these clay minerals or silica, and towards the crater rim. And picking up samples as we go, as some really particularly fascinating samples strike our fancy. Of course, we'll have to collect a lot of imaging data and in situ mineral analysis to understand what these samples really are. And this is the current resolution of all this imaging. So I was saying tens of meters spectroscopically per pixel, not much better, I think, or not even, possibly even worse for just photographs. So we don't really know, as Gale Crater taught us, curiosity. All these minerals, one observes from space, there's much higher mineral diversity on the ground. Once the rover hits the ground and looks around, there's just much more to see, as well as geologically. So what can we do with all this? Just knowing what we know now, or what kinds of hypotheses can we develop? So one of the most exciting in my mind opportunities that we can, or questions we can ask, that we can't ask on Earth just because we don't have sediments this old. There's just no record. There's erosion, there's volcanic activity. We just lose very, very old rocks, or they get cooked and all the signals in them really get obliterated. But on Mars, that hasn't been the case. There was water, there was not so much volcanic activity, and now it's all dry. So we really have a very old surface that is preserved. And that allows us to ask for the first time if there was any kind of prebiotic chemistry, or if we want to be more ambitious, life on early Mars. So what can we look for? So in terms of ages, here's what we know and what we don't know. I said the lake itself is estimated, and this is an estimate, to be about 3.8 billion years old. And here is just really a schematic of what we know about the origin of life. We know that the planets, including Mars and Earth, had a stable hydrosphere starting from before 4 billion years ago. And then somehow life happens. So first, it's preceded by prebiotic chemistry, so chemical reactions that produce a set of compounds that go towards forming information molecules such as DNA, RNA, pick your information molecule of choice. There is some world where there's no cellular life as we know, but these molecules are competing and being synthesized and self-replicating. There have to be some amino acids and there have to be some lipids that eventually first form the first cell. Now, how long this entire period took that led to cellular life, this is really all a schematic. It could have happened immediately at 4.1 billion. It could have happened throughout this time. We don't know, just because we don't really have good samples to ask that question in. And then eventually what we do know is that cellular life arose and formed some of the first sedimentary structures such as stromatolites that on Earth are as old as 3.4 billion years. So all of this happens here. How fast? We don't know. So what would the prebiotic chemistry look like, or what would we look for? There has been some recent experimental work, so no one's been able to create life in a test tube yet. But people have been doing very careful and very, I think, inspired experiments. Some labs have been doing very inspired experiments to start addressing reactions that would lead to a limited set of chemicals that are now present in cells, such as amino acids, such as lipids, and such as nucleotides. And you don't have to worry about this complicated scheme. It's complicated, but it's actually quite simple compared to something that would produce meteoritic or cometary organic matter. So what we have is the property of life that, or prebiotic chemistry that distinguishes it from really the goop that we get from meteorites. If you look at what's present in meteorites, there are tons of amino acids, most of them never used by life. Prebiotic chemistry, in contrast, should produce a very limited set. So really have selection, which is what life does, and self-replicating molecules that then further select. And so this chemistry, this set of reactions that are interconnected, seem to produce a limited set of amino acids, some lipid precursors, and nucleotides. And this has been all experimentally constrained. So the best known, and this is again the beginnings of the field, but the most plausible mechanisms that lead to these compounds seem to, this is important, and this is also why Jezero, as a surface environment, is a great environment to sample. All of these reactions require surface conditions. So to lead to, say, destroy some intermediates and really lead to stable nucleotides, we need UV radiation. We have to have that on the surface. We have to, for some of these reactions, have cyanide, which is thought to be delivered from the atmosphere. So something that is, again, present at the surface. A lot of these steps in these synthesis reactions, so this is done by some very serious organic chemists who really know their chemistry and understand the conditions at which you can get all sorts of precursors. Some of these reactions require products or intermediates to be concentrated, and evaporation is really the most plausible mechanism by which that can happen. Again, we need surface to evaporate solutions. So Jezero, as a surface environment, allows us to look for some organic matter. And then by analyzing it, we can see, is it something that really can be, that's similar to what's present in meteorites? If yes, then maybe all organic matter that is present on Mars was just delivered from meteorites and there's nothing else to it. But if we find some more limited set, that could point to these early steps, because this is an incredibly old environment. And this is all I can say at this point. We still don't know a lot about these reactions, and we don't know a lot about the preservation potential of all of these steps and their different intermediates. And what we do know more about is cellular life. And as I mentioned, early microbial life is thought to have existed on Earth at 3.4 billion years ago. These are the oldest sedimentary rocks that really are undisputed as such. And we don't know much about processes that preceded that life. So we think photosynthesis was present by about 3.4 billion years ago. So we have 400 million years of sedimentary history on Mars that could answer some of the questions as to was life present and what kind of life was there before what we have on Earth. And some of the possible processes that I'll mention, and I'll mention some of these microbes later, are, well, photosynthesis, photosynthetic processes, as well as methanogenesis. This is thought to be one of the oldest metabolisms, particularly methanogenesis that takes up hydrogen and CO2 that could have been present on Mars and makes methane, which is a great greenhouse gas. So we don't really know where, when, and how these processes originated. So what we do know is how the earliest life was preserved on Earth. And it turns out a lot of the types of minerals that are present in sedimentary rocks on Earth are great to preserve evidence for life. So here is really the oldest evidence for life on Earth, microbial life in marine environments. So what you're looking at is a thin section of a rock that is called chert. It's made of amorphous silica. So all of this light stuff is silica, silicon dioxide, and all of these dark wiggles are really layers that are carbon rich. These are carbonaceous. This was some old organic material. And because these layers are bent, you see all these wiggles and curves. It means that this material was soft before it was turned into a rock. And the only soft materials that you can bend in these ways are organic. And this provides evidence. This is interpreted as evidence for the presence of some microbial mats or biofilms. So really organic films formed by microbes this is 3.4 billion years ago. This particular sample is in South Africa, but keep in mind silica, silicon dioxide that could be produced and may be present in Jezero crater is a good recorder of biosignatures. Here is another example of the record of early life. This is, again, much younger, but you can see that this is just to illustrate how beautiful the fossils are that silica can preserve. So this is, again, chert. So this is amorphous silicon dioxide in a lot of carbonate, a lot of dolomite, magnesium-rich carbonate, just like what we expect on Mars. And you see these layers in these dark areas. This is chert. It's dark because it's really organic rich. It has carbonaceous material. When you blow it up and you look at it under the microscope, you actually see that some of these layers contain finer layers that are dark, that contain themselves a lot of remains that look like this. So this is 10 microns. You actually see remnants of former cells. And these are thought to be microbial mats again. So silica, it has tiny, tiny grain sizes and preserves information about cellular morphology really well. And the preservation quality, this is something my lab does a lot. Here's a graduate student who was working. She was really curious about how organisms, how do you turn a cyanobacterium, something that is photosynthetic, into a rock, and how does that depend on any kind of properties of microbes themselves? So what she did was she was growing a lot of microbes and exposing them to solutions that contained silica under different concentrations. And so what you're looking at here were two microbial cells, and they are surrounded by a lot of goop. It's really sticky, and like uh, it's like a marshmallow. It's fairly spongy when you touch it, because these these cells secrete a lot of extracellular polymeric material. It's a lot of carbohydrates. They tend to retain water. They have all sorts of protective properties. And it is these carbohydrates that are around cells themselves that tend to really bind silica well. And how well they bind silica depends on how much magnesium is there, and really the composition of what is in these polysaccharides, these carbohydrates around the cells. So here's just an elemental map to show you that silicon really binds this material that's around the cells. The cells themselves just got degraded. So they don't get preserved, but everything around them, which is the envelope, which gives them the shape, does. So what is cool here is understanding the mechanism, knowing that magnesium really does tend to bind with silica and help silicification is good, because now we know maybe you should be looking for silica that is rich in magnesium. Magnesium is not the only divalent cation that can do that. Here is an experiment that we did in the lab where we grew microbes that don't make oxygen, and we grew them in the presence of a lot of silica and iron too. And it turns out all of these fine granules, you can see tiny, tiny silicon granules that actually, here's a shape of a former cell. The cell itself popped out, but its envelope got preserved. So iron, and you see that the precipitate has quite a bit of iron in silica. So iron can do the same. And remember, both magnesium and iron are present in that source material, olivine. So if you find amorphous silica that's enriched, this could be a good sample to pick up. And some orbital data to tell us that there may be hydrated silica in Jezero crater. So here's our landing ellipse and the delta and the trajectory, and there are certainly areas close to the delta and within the traversable trajectory that could be sampled. So this area here, these are tiny areas, but I'm sure once we land, we'll see a lot more diversity on the ground. There could be areas that are particularly good for the preservation. If there were any fossils or any life, there could be good areas to sample. So that would be, say, a possible exploration route. All right, so I mentioned carbonates as well. These are, well, on Earth, carbonates form reefs. This is what forms all limestone. It's really the entire coast of Croatia. That's what it is. Although the carbonates on Mars contain a lot more magnesium instead of calcium. So it's not really limestone, it's magnesium carbonate. And here is the orbital data that shows where these carbonates are. And recently they've received a lot of attention, because when you look, so here is what you should be focusing on here. So here's a lake, this is the crater rim. Everything that is here in blue or purple contains quite a bit of carbonate. And it seems like these carbonates, so these blue areas, are distributed close to the delta. And within, so here is what these researchers did. They mapped out the topography, and they showed, so all of these lines are lines that record the same levels of former levels of the lake. And they found that the carbonate distribution is really restricted to the high stand, former high stand, where the water was the highest of the lake, and don't really follow the lake. So it seems like really a bathtub rim, something that indicates or is consistent with mineral precipitation as perhaps the lake level starts dropping. So you still have surface waters that are in contact with the atmosphere. There's some evaporation, and this is where minerals form, just like what you have to clean up with vinegar in your bathrooms. And carbonate minerals are great as recorders of temperature. If we have samples back on Earth, we can use these regardless of how they formed. We can use them as isotopic recorders of paleotemperatures, and this is what people use them to reconstruct past climate. They can use them as records of water levels as well as records of water chemistry. So magnesium carbonate, it's MgCO3. That carbon comes from CO2 that could be in contact with the atmosphere. So we can also say something about the former atmospheric composition. So carbonates, if they look something like these are lacustrine carbonates in Walker Lake. So here is, that's in Nevada. This is a lake that had a higher water level in the past, and when it was higher, these carbonates were really deposited along the rim, and people study them exactly for the information that is listed here. So temperature, water layer. So these carbonates seem to track the former water levels, and they have different shapes just because of different physical conditions that were present and chemical conditions as the lake was changing. So ideally, we would see something like this. The question is still, what are these carbonates? Because like everything on Mars, there is a lot more confusion and there are a lot more potential sources, including some delivery of carbonates from areas that surround the lake. So we still, one of the really zero order questions is, what are these? Why is there more carbonate in this area? And what will we see? But if we see something like this would be ideal to sample. Another reason to sample carbonates is that a lot of the evidence for past life in shallow water environments on Earth comes from carbonates. And here are some examples of magnesium carbonates. So here are stromatolites. These are 3 billion years old. These little conical structures, and they have also a lot of silica. They have a lot of magnesium. And there, if you cut them up, you see textural evidence that these stromatolites were made by microbes. There was some former sticky surfaces that were shaping the rock. This is a very unusual shape. So, if we are lucky to see something like that, we just sample it and call it a day. That's it. It would be a very successful day. This is not necessarily what we can hope for, but the neat thing about microbes on Earth, at least, is that they can stabilize magnesium in carbonates. Magnesium carbonates really they're kinetically hard to make. So if you just have solutions that are not evaporated, you will have a really hard time making magnesium carbonates just in normal surface waters. But microbial, so what you see here is a lot of microbial cells. So here is these are all cells that tend to, because of their surfaces, bind magnesium and actually stabilize magnesium-rich carbonates. So the composition of the carbonates, depending on the broader geologic context and whether they were just some evaporate or not, can also tell us or point to samples that may have required microbial stabilization. Iron can also do the same. So microbes can stabilize iron carbonates as well. Okay, so these are major minerals produced by aqueous activity. Another class of minerals that really forms the delta, is really abundant in the delta, are clay minerals. And don't worry about too many different colors, but again, colors here map the distributions of different types of minerals. And so to some, this is just really a replotting of similar data, just focusing on different absorption bands. So we have a red-yellow is magnesium or iron clays, clay minerals, again, that require interactions between igneous minerals and water to form, or carbonates. So these are, again, tricky sometimes to distinguish, and that is that carbonate-rich area, but there are some places within the delta where both are present as well. There are aluminum clays, so there is some aluminum present. There is silica, and so on. So the clay minerals really form the physical delta. That is what's in the layers of the delta. That's what was rolled around as the river was discharging into the lake. And clay, deltaic deposits and clay-rich deposits on Earth are great to look for microfossils in, because if you have a lot of fine-grained minerals that dump onto any kind of microbes, they protect them from the surface, they protect them, they cut off the delivery of oxygen. And so here are some experiments that we did, and microbial surfaces are actually great at binding them and becoming fossilized. So this is a former PhD student of mine who was doing experiments with microbes. So these are these filamentous microbes. These are all sorts of cells that that have sticky surfaces, and they just form aggregates. If they form aggregates in the presence of clay, that can come from anywhere. You see this Y, for example. This was one filament, this was another microbe, and these clay minerals just form fine coating and they preserve the shape of the microbes. And in fact, if you were to remove the clay minerals, you would see even beautiful organic envelopes. Again, it's always these sticky envelopes that are preserved. This is just to show you that these have aluminum and silica, some iron and magnesium. So clay minerals are great if we can recognize some quite rapid deposits, and we can look for biosignatures. Another type of potential information, or information that is potentially preserved even in Jezero crater, is hinted at by some observations from Gale Crater. Again, just huge evidence of interactions between water, maybe some atmospheric components such as CO2, and the Martian minerals. So these are images from Gale Crater, but features that we haven't really seen before or didn't really know what to make of that we could expect in Jezero crater. So when Curiosity rover landed in Gale Crater, it started looking at some of the first deposits that were mudstones, so really fine-grained clay minerals. They're also enriched in silica. And yes, you may be used to the Martian surface looking red because of oxidized iron, but as soon as the rover started drilling these cores, people noticed that under the surface, very shallow surface, there's a lot more gray, meaning this iron is less oxidized. There was less exposure of that iron to oxygen or nitrate or whatever can oxidize it. And these gray layers are really enriched in magnetite, and some of them contained features. So this was done even without drilling. You can see it with the rover cameras. Some of these layers of this fine material contained these granules that are millimeter sized. So these are little blebs that formed in sediments as the sediments were deposited. So really early on in the history of sediments, some of these blebs are filled, some of these blebs are hollow. Here is another picture. They're really layers and layers of these blebs. Here again, more layers of blebs and ridges that come really right across. And this is in this basaltic. So the original composition of the sediment was fine basalt that somehow interacted with water. We can see that from the sedimentology. And the question is, how do you make it? This is not something we've ever observed on Earth. And the most plausible mechanism by which people, Mars is full of mysteries, but the only plausible mechanism by which people can explain the formation of these really regularly sized features so these are also 3.5 billion years old, is that you have some gas that forms bubbles within these fine-grained sediments, and these bubbles get cemented and their shape is preserved by that process. And sometimes you get bubbles, and sometimes you get probably just gas emanating from broader areas, and you get the ridges that are raised. Everything happens really early on in the history of the sediment, meaning whatever these cements are, record aqueous really reactions between water and minerals right then in the sediments. And because these bubbles can be preserved, I mean, not the gas itself, but the shape, something has to form, some solid phase that will preserve that shape. And that solid phase, because it forms really early in the history of the sediment, should be able also to preserve organic material, if any were present. And so here's the exciting part about the bubbles, or just these features. So if you start wondering what kind of gas has to be there, CO2 cannot really emanate from sediments unless you have a lot of organic material, which would be good news. The only gas that we can make abiotically on early Mars, or imagine abiotically, could be hydrogen, which is a lot of hydrogen coming from fine basaltic sediments. And we don't know what the cements are. They are rich in magnesium, they are enriched in silica, they could be amorphous, but we think they would be really good candidates. If you see any features like that, that would be great to drill and collect. What's exciting about hydrogen, we don't really know mechanisms by which it should form, just if you have water reacting. But if you have a high PCO2 atmosphere, so you have a lot of carbon dioxide in the atmosphere, which is thought to have been the case on early Mars because we needed liquid water and we needed a lot of warming to maintain that water liquid in lakes. Then hydrogen can act as a greenhouse gas and add additional warming. So this could be almost a geological mechanism to keep Mars warm, at least for a short while. And so that brings me to the last part of how, when, and why did Mars change? 4 billion years ago, there is no doubt that Mars was warmer, which is not hard because today's surface temperature is lower than minus 60 degrees, the average Celsius, and wetter. This still can mean that most of it was under glaciers or snow, and there was just a little bit of water flowing. But today, unambiguously, it's cold and dry. So what happened? One of the major ideas is that its dynamo just shut off. And the early dynamo, the idea, these are all, so here we are now in the realm of physics and uncertainties because there are all sorts of models with different predictions. But the idea for the early dynamo is that it would protect Martian atmosphere from the solar wind. And if you turn it off, then the solar wind comes, impinges on the atmosphere, kicks off gas molecules, and Mars just loses its CO2 and other gases and becomes very cold. So this is what happens when there's no core dynamo. So the question also we could ask, if we find rocks that contain some magnetite, is that, which if you remember, I mentioned there's quite a bit of magnetite around those nodules and ridges, those bubbles. If we get rocks that have some magnetite or some mineral that can record magnetic field, we can start asking questions, especially if we collect multiple rocks along the trajectory that reflect a time sequence. We could start asking where the Martian dynamo turned off. So what are some of the possible reasons for the climate change? And the way we know that Mars had a dynamo is from meteorites. Here is the oldest Martian meteorite, ALH 84001. This rock is 4.1 billion years old. So it landed on Earth. It landed in Antarctica, Allen Hills. That's really what the name tells you, ALH. The rock itself is 4.1 billion years old, but we don't know where it came from. So this is the advantage of having rocks back that come from a known geologic context. We don't know where on Mars this rock is from, but what we do know is that it contains magnetic minerals that were magnetized in the presence of a dynamo. So we know that Mars had a dynamo at about 4.1 billion years old. So 4.1 billion years ago, or maybe somewhat earlier. When the dynamo turned off, we don't know. It could be right around the time of the river network activity on Mars, maybe somewhat later, maybe even earlier. But having samples from Jezero crater could help us start understanding this timing. So here is really this entire period, this is what we know, this is ALH, this bar here. All of this is in question. It could have, the dynamo could have turned off at any point here. So another great way for samples from Mars to answer first order questions about really planetary evolution, of the evolution of our sister or brother planet. So here is a possible exploration route at Jezero. So if you start from the floor, this flat area, go over the river and sediments, not only can we acquire samples that allow us to maybe date them, at least the surface exposure, and say something more about how long this lake even lasted, which is another uncertainty. We can keep sampling, go towards the crater rim, and eventually, if the mission lasts, exit the crater and really date the terrain that is above. So really get the whole temporal sequence of events that shape this environment, which would be, again, unprecedented, dating a sample with terrestrial instruments that allows us really to, it would allow us for the first time to constrain and calibrate entire Mars chronology. So here are the conclusions and some plans. So Mars 2020 and the Perseverance rover will begin the Mars sample return campaign. It's an entire campaign. As I mentioned, the rover is the first step, the kickoff. This rover will pick up samples, select them, and cache them. The subsequent steps of this return campaign will come and pick up samples and bring them back to Earth. All of this is in development now. So this will require collaboration between NASA and ESA. ESA is a major player, the European Space Agency. And the samples, it's again not quite clear when they will be returned, but some optimistic estimates had them as back on Earth, had them on Earth as early as 2028. And these samples can tell us a lot about a lot, including the age of Jezero crater and the timing of really the water and sediment deposition in it. They can tell us a lot about the history of Martian climate and magnetism. They can tell us if we see any organic matter. They can tell us about how that organic matter was produced. Was it delivered by meteorites? Was it produced in situ? Was it maybe produced even by biology? It can tell us about the entire carbon cycling on Mars. And it can tell us about potentially, this is the most exciting, also the most tentative part. If we see anything that resembles a biosignature, then I think that can spark a whole lot of studies and debate about the early biological evolution, as well as prebiotic chemistry that led to life. Just because we have these incredibly old sediments. So I will end here in terms of the main presentation. The rest I can discuss or maybe take questions now, and I can tell you about some other experiments to really get at Mars-specific processes that will enable us to recognize cool samples that we don't have on Earth that could record evidence of past life or really just interactions between water and the atmosphere.